displayed for you what does it mean in terms of how similar two individuals are with a correlation coefficient of about 0.8. They're not identical, but they're quite similar. And it happens that I have chosen a case of two individuals, one from Spain and one from Denmark. Isn't it extraordinary that they'll be quite similar? Now, let me go through the color code. Obese individuals are in red, overweight in yellow, lean in green, and the size of a circle reflects their BMI. So if you look at this network, which links about 20 individuals, you can see that in one part, they're mostly lean. And the other part, well, they're interspersed. So maybe one should try to analyze, compare their microbiota, not across the whole cohort, but trying to choose individuals which are more similar. And that brings to mind the concept of enterotypes, which is like, blood types or tissue types. But it's quite early, anyhow. If one lowers the threshold from about 0.8 to 0.7, one gets a network which is quite complex. We need to learn more. There are some subnetworks. There are regions of the network which are less dense than the others. The situation is more complex than when one looks at the very top, but it's kind of intriguing that, there seem to be quite high correlations between individuals, but some are much more correlated than the others. I can stop here. Before addressing functions of the encoded by the reference gene set. Okay. 3.3 million genes don't encode 3.3 million functions. They can be grouped in clusters, and one can examine the number of clusters that we have detected. There are about 6,000 of functions which were previously characterized, and about a double of previously known functions when one pulls well and poorly characterized functions in function of number of individuals that were sampled. And see how we saturate. We think that we have seen most of the functions. When we look at all functions, not only previously known, but also the ones that we have detected. Okay. We have identified very many of these new functions. I will not take time to go into minimal metagenome functions that are absolutely conserved, but would like to switch very quickly to the end because I do need to acknowledge many people. Maybe just flash this thing for you. We can distinguish quite well healthy people from ulcerative colitis and Crohn's disease people. This is very exciting, but quite preliminary. And the take-home messages is that we have means to constitute genes in metagenomic analysis by short reads. We have identified 3.3 million prevalent human intestinal genes, 150 times more than human gene complement. This gene catalog includes most of the genes identified in the studies over 3 continents. And we think that we are facing the human metagenome. A number of people helped MetaHit Consortium. Most importantly, people from BGI, EMBL, and a number of other people, Soren Brunak, Joël Doré, Francisco Greatly, with all of Pedersen clinical colleagues, sequencing centers, and people from my own unit. Thank you very much.
Well, very interesting and fascinating talk. I wonder whether the metagenomes of animals are also known, and whether what kind of relationship is there between the human metagenome and other metagenomes or animals, if anyone knows.
Thank you for the question. It is an important question which I haven't addressed. We have projected the reads from the mouse metagenome on our contigs, and very few map. Genes present in mice are quite different from the genes present in humans at the level of threshold which we require. We have not gone systematically over animals, because for that you have to create metagenomic sequences, which we do not have. And 16S comparisons are simply not good enough for that. But for very important animal, experimental animal, mouse, we do have an answer. And I think that the corollary of that is that one needs to be careful when extrapolating results from rodents to humans.
So, impressive talk. I think it's really surprising to see that enterotypes are not related to the geographic origin of the subjects, but are just that widespread all around. But are you thinking to try to do some kind of control beside this huge amount of information you're collecting that needs huge informatic means and for sure a lot of money, to check in smaller cohort of patients to try and see, or people, let's say, to try and see if the correlation, I mean, because it's difficult to understand if these differences in the metagenome, so in the population of bacteria that are in the gut of different individuals, can be a cause or a consequence of certain phenotypes. So, for example, it was impressive, the last correlation you show, where Crohn's disease and other pathologies score differently from the normal individuals, let's say. But could be that differences in the metagenome are a consequence of the different activation of inflammatory pathways in the individuals that you are analyzing, or in a different immunological response that each individual is able to produce against that particular kind of bacteria, you know? So how can you check that?
Thank you for the comment, which is absolutely relevant. I thought I was quite careful in my talk and mentioned associations and no causal relation. But the first thing is to try to detect associations. Once you have associations, you can try and study further and try to determine what is the cause. And basically the function analysis part of MetaHit is kind of, primed to go and try to understand the mechanism if there is one. Now, what I should say before Francisco intervenes, because he can comment much more knowledgeably about IBD than I can, is that all individuals in this study were in clinical remission. They were not under any treatment, antibiotic treatment. So, all I can say is that that's the difference that we seem to have based on nevertheless, a very small part of our cohorts. And before I really trust what I have projected, I think we need to just a little more patience and, but on the other hand, it's so exciting, that I couldn't resist. I know that I shouldn't have projected it, but I couldn't resist.
It was nice, though.
Let me make a comment. I think it's quite interesting, because what happens is the study design is that we enter patients in remission, which is patients with only might have 5-ASA treatment, which is oral therapy, not absorbable drugs, so all the patients in the same situation. And we follow them for 1 year to see what happens when the patient starts a relapse, so when the disease becomes active again. And in this study, you have the controls, the family controls, and the patients in remission. And it's quite impressive how they differentiate, even if they are in remission. And it's quite impressive how the metagenomic approach established that really they have a less diverse genomic functions in these patients. They might explain why they are so unstable and they develop bouts of disease. I think it's a quite interesting finding, even if it is preliminary, but yeah, I think it's
I feel so good, not only because Pancho approves, but also for once I can look at him from above.
Could you envision that there will be maybe a sort of metagenomics diagnostics in the future, so that you don't know exactly the kind of disease in a given patient, but you screen the metagenome and could say, that's it?
Well, that's one of the goals, not direct goals of MetaHit, but we do hope to identify bacterial signatures of diagnostic and prognostic value, and that should be converted into tests. Now, one of the last partners that joined us, bioMérieux, are one of the major players in bacterial diagnostic all across. So I'm extremely pleased that they are with us, even if they are involved in one part of the study which is not directly related to their thing. But you are absolutely right. You know, when I dream about the future, I say, when I go and see my doctor today, he'll send me for blood test, for urine test, but maybe in 10 years, he'll systematically send me for fecal test, because that will open a whole new window into my physiology, my pathology, and maybe the next step of a dream is that maybe we could use foods, very mild treatments, to prevent rather than to cure. And if we have to cure, that we will have a panel which will tell us, why do we treat this guy with this? Maybe it would be better to treat him with something else. So it goes to the personalized medicine in Metagenome. So that is kind of vision that one may have. And I am very pleased to say that the number of centenarians in France is increasing. So, at my age of 65 and 35 years of career, I have still 35 years to look at. I hope I see that.
Thank you.